Hello everyone, a very good evening to all of you. Today we begin with the second session on science and technology as a part of our Target Prelims 2023 sessions. And today, as we did yesterday as well, we will be discussing some of the key areas from the perspective of science and technology, especially giving a focus on current affairs of last 1 year. So yesterday we discussed some of the key areas, we discussed some of the areas of importance. For example, we discussed about the various aspects or various news articles related to space technology, related to artificial intelligence and related to intellectual property rights. So these were the three topics from where we discussed some of the key questions which have appeared as a part of the news in the last 1 year or maybe last couple of years. And today we'll be just carrying forward from where we left yesterday, and we will be discussing three more very important areas. First of all, starting with biotechnology, we'll be discussing some of the key areas of biotechnology. We'll be discussing some of the questions related to all these news. Then after that, we'll also move to IT and communication as our second topic. We have picked some questions from there as well, and towards the end of the session, the last few questions will be related to the Nobel Prize of last 2 years. Nobel Prize of 2021 as well as Nobel Prize of 2022. So these are the topics that we are going to discuss in today's class, and these are the topics from where the questions have been picked in today's session. So let's begin with the session. Let's begin with question number 1. Now, so question number 1. So question number 1 starts with biotechnology. So as a part of biotechnology, what we see here is, consider the following statements about DMH11, that is GM mustard in India. First, GM mustard will have beneficial effects on honeybee population. Second, Australia has recently released herbicide tolerant GM Indian mustard to meet the increasing global demand of edible oil. So these are the two statements. You have the options in front of you. We are launching the poll for you. Please mark your answers and then we shall have a discussion on this particular question. So just about to launch the poll for you. Yeah. So please mark your answers and let's have a discussion on this topic. Alright, so as I see, many of you have marked your options. I'll just wait for 10 more seconds and then we shall start our discussion. Alright, so let's start with our discussion now. So first and foremost, what are we discussing? We are discussing GM mustard, and we are discussing the Indian variant of this GM mustard, that is DMH11. Now this DMH11 has been in the news on and off in the last 5 years, and that's why it becomes a topic which is important. There is already one question that UPSC has asked from here, and that was when for the first time the discussions were happening and all the debates were happening around this GM mustard or DMH11. So let's try to understand first of all what is this DMH11, and then after that we shall have a discussion on what are the various aspects of this that we need to understand. So first and foremost, what is this DMH11? So DMH11 is a hybrid variety of mustard. Just remember this, hybrid variety of mustard, and a hybrid variety that was supposed to be released for the Indian farmers back in 2018. And this is where it was also cleared by the Genetic Engineering Appraisal Committee. It had already given its go ahead, and then after that the center was ready to launch it for the Indian farmers, and this is where there were some concerns that were raised against this DMH11. And because of the concerns that were raised, these concerns were from some scientists, some environmentalists as well, and some farmer organizations also. Sorry. So because of this, some concerns that were raised, it was also taken to the courts, and when the matter was taken to the court, this is where a discussion happened and the court has put a stay on this release of DMH11. There were certain reasons. The basic idea that was proposed back then was that let's do further studies, let's try to understand each and every aspect, let's try to see and ensure that there are no negative aspects, there is no issue that this DMH11 would cause, whether with regards to human health or with regards to the environment. So these are two basic aspects that were raised, that it might have some negative impact on the environment or it might have some negative impact on the human health as well. So these were concerns that were raised, and that is why the court had taken its cognizance and they had started discussing this out. And then this is where we also saw that a stay was put on DMH11 and a committee was formed. And this committee that was formed, this committee was given a task that you start studying DMH11, look at the various properties of DMH11 and come out with a report to understand whether or not there is any issue that might be caused because of DMH11. Any issue, whether environmental, whether on health. So that happened. Then after that, coming to 2022. In 2022, this committee comes out with its report and says that they did not find anything problematic, they did not find anything negative, and they feel that now it is ready for the release into the Indian markets. It has not been released yet, still has to be given a final go-ahead from the side of the government, but we see that this is what the status is. So this is what we see with DMH11, that first it was about to be launched, and then we see that there was one stay that was put here. Now, when we talk about this DMH11, the aspect of the fact that okay fine, a stay was put and there were some concerns that were raised, first and foremost we need to understand why exactly was this DMH11 being released. What is the advantage that DMH11 gives us? So when it comes to the advantage, the basic advantage is productivity. The basic advantage is productivity. The yield that we see with DMH11 is very good, that even compared to the best of the variants of mustard that we have in India, compared to them even, we see that DMH11 gives a better yield. For example, some of the best variants like Varuna, for example, it was cross-referenced against Varuna, and when the discussions were happening here and when the research happened here, when the studies happened here, they found that it has almost up to 25% better yield as compared to the Varuna variety, which is supposed to be one of the best varieties of mustard available in India. So even compared to that, it was showing a better productivity. So that is where we see that DMH11, all those studies that happened over a decade basically, they finally were about to release it into the Indian market. Now, the concerns. We are talking about two aspects of concerns. One, from the perspective of health. Second, from the perspective of the environment. So what are these concerns? What are the concerns, for example, with regards to the health? Very often you'll see that whenever there is any food crop which has some kind of genetic modification in it, we see that there is a lot of opposition to it, because we need to understand that whatever genetic changes you are doing, whether those genetic changes could have a negative impact on human beings. Can it have a negative impact on the human beings? Although most of the studies that have happened until now in the world, they haven't shown anything negative, they haven't shown any negative impact, but the concerns remain that most of the studies, for example, which are being done, they might have been done over a span of 5 to 7 years of time. We need to understand that can it have an impact, let's say 10 years later, 15 years later, 20 years later? Because if it has a negative impact 20 years later, it would be too late for us to even realize that there was an impact that it had. So that's why we want to be very sure that whenever this kind of a gene modification is being done or any kind of gene editing is being done, is it going to have any negative impact on human health? So that is why it was said that we should have a better discussion, we should have a much long period study, and once we have this study done in its place, then only we should move forward with the release. And this is basically what was said back then. So now, this was about health. Second was about the environment. There was one problem that was raised, and that problem was also with regards to the biodiversity and the effects that it can have on biodiversity, the negative impact that it can have on biodiversity. So for example, when we consider mustard, there was this point raised that there are 66 variants of mustard that we grow in India. 66. So once you have this genetically modified mustard also available, and it has a better yield as compared to most of the other variants of mustard available in India, then it also means that the farmers might stop growing all these other variants, and they might start moving towards this DMH11. Then what happens next is that it also starts to lead to a decrease in biodiversity, because many of these seed variants which are being grown in India, they might perhaps will not be grown anymore. They might not be liked by the farmers, and farmers perhaps will not grow them anymore, and that is one of the issues that it might cause. So then ultimately it can lead to a decrease in biodiversity or species diversity of this mustard variant. So this is one problem that was raised. Apart from that, there is another problem, and this is what the first statement is about. GM mustard will have beneficial effects on honeybee population. It's the opposite of that. What many believe is that it might have a negative impact. It might have negative impact. Remember, I'm not saying it will have. All right, I'm not saying it will have. I'm saying it might have a negative impact on bee population. Now, where is this concern coming from? All right, so there are 2 points that have been raised when it comes to honeybees. So first problem is that, let's say when this mustard is being grown, one reference that they have, the reference is also from BT cotton. That when BT cotton was released, when BT cotton was introduced, this was also when there were some concerns that were raised, that because BT cotton, when you talk about this BT cotton, what we know about BT cotton is that this BT part is going to act like a toxin, right? It is going to be poisonous. Poisonous for what? It is going to be poisonous for the pests. What kind of pest? Basically bollworm, right? So we know for sure that for bollworm it is going to be poisonous. But apart from that, there are chances that it might become poisonous, or it might have some kind of negative impact even on the bee population. So there were these concerns that were raised. Although nothing very substantial was found, most of the studies showed that it would not have any negative impact, no positive impact anyway, but no negative impact. So most of the cases, but yet it has been raised as a concern that if there is any genetically modified crop, we should do these kind of studies. So this was one, that maybe because if they go to mustard flowers and they are going for pollination from there, it might have a negative impact on the bee population. So this was one problem that was raised. Apart from that, the second problem that was raised was with regards to that, can it have an indirect impact on honeybee population? How can it have an indirect impact? Understand this, that this indirect impact was also raised as a concern against BT cotton. That in case of BT cotton, because we are saying that BT cotton is able to produce its own pesticide. Basically, producing toxin means this, that it is producing its own pesticide against bollworm. So it means that you don't need external pesticides, right? You don't need to use pesticides externally against bollworm. So what then happens is that the use of pesticides drop, which is good, but at the same time, because of this decreased use of pesticides, what may happen is that some of the other insects which do not have any negative impact as such, let's say on cotton, but they are there in those areas, they might start to increase in their population. So if their population increases, then in that case there is a competition. There is a competition for pollination, there is a competition for acquiring food from various sources. So that might have a negative impact on honeybee. So that's why it may not have a direct negative impact, but it may have an indirect negative impact, is what was also being discussed at the same time. So that's why we are talking about these situations. That's why we are talking about these issues here. So that's why remember that they might have negative impact. There are chances that it might have a negative impact on honeybee population, and this was also one of the concerns that was raised. And again, when the release is again being talked about, we see that this concern is back. So that's why this concern is what we are discussing. So this cannot be correct. Second, Australia has recently released herbicide-tolerant GM Indian mustard to meet the increasing global demand of edible oil. This is also correct. It happened last year, I think, in the month of October. This is where it was done by Australia, that they released a genetically modified mustard variant, which is a kind of an Indian mustard seed. So now that mustard variants are being released, this is why it was again raised for India also, that if Australia has released it, India should also release it. India should also move forward, because India also has this one major issue, one minor issue I would say, that we also import some of our mustard. Our mustard requirements are not completely met indigenously. Alright, there is some mustard that we also import. So that's why, because of that also, we want to increase our productivity, because if we are able to increase the productivity, this is where we'll probably not be dependent a lot on our imports. So that also is one of the concerns that have been raised. So what is correct has been asked here. So the correct answer will be 2 only. Alright, correct answer will be 2 only. So just remember the context more than anything, because why DMH11 again is in the news, because now we are starting with the release of GM mustard, or we are again discussing the release of GM mustard. Alright, so let's move on to question number 2. Consider the following statements regarding the chimeric antigen receptor T cell, that is CAR T therapy, that was in the news recently. Clinical trials under the therapy conducted globally have shown promising results in curing final stage cancer patients. The therapy is cost effective when compared to other treatments. And third, it involves genetic modification of patients' autologous T cells. Alright, so these are the three statements given to you, and you have been asked what is correct here. So please mark your answers. Option D is not visible. Some of you are saying option D, if it is not visible, it is all of the above. Alright, option D is all of the above. So please mark your answers and let's have a discussion here. Alright, I think most of you have marked your answers. Yeah. Alright, so let's start with our discussion. Let's try to understand what exactly it is. So first and foremost, when we are talking about this therapy, the CAR T therapy, let's try to understand first of all what do we mean by this therapy and where exactly are we using it. Alright, so just try to understand the basic process here, that what do we do with this CAR T therapy. So first and foremost, what we are doing is that from the patient's cell or from the patient's body, we derive some blood, and from the blood we basically derive the T cells. Alright, now when we talk about the T cells, I'm sure you know about the T cells. What are T cells? T cells are basically cells which help in the immune response. So these are cells which are associated with our immune system, and they help in providing the immune response against any incoming antigen or any kind of antigen in the body. Alright, so this is basically what we see here. So now in these cases, when it comes to T cells, what we are trying is that first of all we are getting the T cells from the patient's body, and because we are getting the T cells from the patient's body, that's why we call it as autologous. So when it comes to this particular term autologous means that when you are deriving something from the body of the patient itself. So then in that case it will be called as autologous. So autologous, wherever you hear the term, just remember that autologous means that we are talking about uh, taking the cells or taking the blood or taking the gene, taking whatever we want from the source or from the patient itself. Then in that case it is called as autologous. So here, because we are deriving it from patient's body, that's why we are saying is that these are autologous cells. Now in this particular case, what happens is that when it comes to this T cell, once the T cell has been derived from here, what we insert is a particular gene. Alright, there is a particular gene that will be inserted here, and this particular gene, when it comes to it, understand what this gene is. This gene is called as CAR. Alright, CAR, or what we call as chimeric antigen receptor. So this chimeric antigen receptor gene will now be inserted in the T cell. Alright, so now T cell basically is being genetically modified. So T cell has been genetically modified. Now these chimeric antigen receptors, basically they are receptors that will be there on the T cells, and now we produce lakhs of copies, alright, thousands to lakhs of copies of this genetically modified T cell. Once it has been done, this T cell or the modified T cell will be introduced back into the patient's body. And once it has been introduced back into the patient's body, this is where we see that the T cells will basically start to lock against the cancer cells. Alright, so the antigens that we have in the cancer cells, what we'll see is that these CAR T cells that we have, the CAR receptors that we have added, they will just go and lock themselves against the cancer cells. And once they have done that, this is where they'll start destroying all these cancer cells as well. So this is what they will do. So and what we are doing is that, again, when it comes to this particular therapy, first and foremost we are talking about adding these receptors to the T cell. So there is a genetically modified T cell that you have here. So these T cells, these are genetically modified. These are genetically modified. How are we modifying them? By adding chimeric antigen receptors. Alright, so by adding them, we have genetically modified them. We now produce lakhs of copies of them, and once the copies have been produced, these T cells will be infused back into the body of the patient, and this is where the T cells now will have these receptors that will go and bind themselves, lock themselves against the antigens of the cancer cells, and they'll try to destroy the cancer cells by doing that. And because it is being taken from the body of the patient itself, that's why we are also calling it as autologous. There are two terms you should know. One is autologous, when you are deriving from patient's body. Deriving from the patient itself. And then the second term is allogenic. Second term is allogenic. So allogenic is when you are taking something or transferring something between two organisms. Alright, so then in that case, between two individuals, then in that case it will be called as allogenic. Alright, so transfer between two individuals. Remember this, of the same species. Then in that case it is called as allogenic. So allogenic would be when, let's say, blood transfusion is happening and the blood is being taken from somebody else, or the gene is being taken from somebody else. But when it is taken from the same patient, then it is called as autologous. So just understand these basic terms. Alright, so coming to the question and what the question is asking. The first is, clinical trials under the therapy conducted globally have shown promising results in curing final stage cancer patients. This actually is correct. And that is why we have seen that in many of the advanced cases of cancer also, this CAR T therapy is showing some promise. Although there is some kind of instability also, it's not that it is 100% perfect right now, but we see that it is something which is getting the popularity because of the fact that even in advanced stages also, it has started showing promise. So that's why this is correct, that it is happening. Second statement is the therapy is cost effective when compared to other treatments. This is wrong. It is not cost effective right now, because a single therapy could, right now, let's say in terms of Indian rupees, it could cost up to 3 to 4 crores. All right, so up to 3 to 4 crores could be the total cost of this therapy right now. So that's why we, in any way, we cannot say that it is cost effective. So that is one of the concerns, and this is where it has been in the news also, because there was one trial that was conducted in India, and it has been almost 1.5 years now. It was conducted in India, and then after that the government also has started working towards it, so that we can create an environment where these kind of therapies can first of all come in India, because right now in India it is not being used. Secondly, also to start thinking of ways to make it cost effective. So that is what we have been trying, and that is the reason why it has been in the news. So, but this is wrong. Third statement involves genetic modification of patents autologous T cells. This is correct. So this is what has been asked, that what is correct. So correct statement will be C. 1 and 3. These two statements are correct. So cancer treatment, because when it comes to cancer treatment, we know that cancer treatment right now has been a subject that is very often being talked about, because when it comes to cancer treatment, the first problem that we face with cancer is that we don't know the exact cause. We know about certain factors. We know about certain factors that can lead to cancer, but we cannot guarantee that every time they will lead to cancer. For example, let's say chewing tobacco. We say that it leads to cancer, but it might not. All right, it might not lead to cancer also. So that's why there are some issues that where we see that there is no exact reason, or there is no exact pathway that cancer follows, and that has been one of the concerns, because of which we haven't been able to find out the way to cure cancer. There have been some improvements that we have seen. There have been some therapies that have been helpful, but again they have not been 100% helpful, and especially in the advanced stages they haven't been. If there is an early detection, there are still better chances of curing it, still better chances of using some kind of treatment. But if it is in the advanced stages, we see that it becomes very, very difficult to handle cancer. And that's why there are so many new therapies that we see with regards to cancer. So whether it is about the CAR T therapy, whether it is about the stem cell therapy also, where we see that some amount of success, some individual cases of isolated cases of success has been observed, yet we see that there is a long way to go. Right, so let's move on. Question number 3. Consider the following statements regarding site-directed nucleases, SDN1, SDN2 and SDN3. These are gene editing techniques used in modern biotechnology for addition, deletion and modification of genes. Recently, Ministry of Environment, Forest and Climate Change exempted SDN1 and SDN2 categories from concerned biosafety regulations, potentially helping in the faster development of new varieties of crops. So these are the first two statements in front of you. Please mark your answers and let's have a discussion on this question. Good to see that many of you are also discussing the topics among yourselves. Great to see that. And please get that discussion going regarding the questions that we are talking, regarding all the discussions that we are doing. That will really help all the others also who are asking those questions. Yeah, so not all the cases of lymphomas. There are only certain cases where we have seen some success with CAR T therapy. So that's correct. Alright, so let's start our discussion. So we are talking about SDN1, SDN2 and SDN3. So let's try to understand. Again, let me just go back to the basics. I know the first 3 questions that I have today, these are bigger topics which you need to understand well so that you can answer any question if at all it is asked. Otherwise, they become highly technical in nature for anyone to understand on their own. So let's try to understand this first, that what do we mean by this SDN1, SDN2 and SDN3. I'm just trying to make it as simple as possible for you. So now these 3 techniques, what we are trying to do with these 3 techniques is some kind of gene modification. Now what kind of gene modification? When we look at SDN1, SDN2 and SDN3, let's try to understand the basic difference that we have between SDN1, 2 and 3. So first and foremost, when we are talking about SDN1, what we see in case of SDN1 is that there is gene editing being done here in the gene itself, and we are not trying to use anything external. We are not taking any template which is coming from any external source. So we are completely confined inside that gene itself, inside that organism itself, and there is nothing foreign here. All right, there is nothing foreign being introduced here. Largely we have seen that there have been 2 gene editing tools that have been used, ZFNs and TALENs. Alright, so ZFN and TALEN, these 2 have been 2 primary genetic engineering tools that have been used in the case of SDN1. Now, these 2, when it comes to it, so these 2 primarily have been used in SDN1. So in case of SDN1, whatever editing is being done, it is being done inside the gene itself, in that given gene itself, and all the editing will be carried out without the help of anything foreign. So this is what we are doing in SDN1. The procedure altogether is a little more complex. It's not very easy when it comes to SDN1. So SDN1 as a concept, or as rather a procedure, has been a little difficult to carry. It has been a little complex to carry out. So that's why we have seen that this is one major problem that we have. Then after that, when it comes to SDN2, SDN2 is where something which is very famous today, very popular today, CRISPR-Cas9, if you have heard of it. Already UPSC has already asked 2 questions from CRISPR-Cas9. So CRISPR-Cas9 is something which is more often than not used in SDN2. Alright, in case of SDN2 technique, we see that this CRISPR-Cas9 is used. Now, when it comes to CRISPR-Cas9, what we see is that CRISPR-Cas9, why has it become popular? Because it has made the entire procedure much simpler. Compared to ZNFs and compared to TALEN, we see that these 2, although were also effective gene editing tools, but they have been very complex. Compared to that, when it comes to CRISPR-Cas9, CRISPR-Cas9 has been much more simpler, much more easier to use. So that is what we see. Now what does it do? We talk about this term homologous template, that it uses a homologous template to carry out the gene editing. Now the gene editing is still being done without the help of any foreign DNA, that there is no foreign DNA that is being introduced into the genetic makeup. Alright, but we are using a guide RNA. We use a guide RNA in case of SDN2. And this guide RNA, when we talk about this homologous template, this guide RNA becomes very, very crucial from that perspective, because it helps there. So when it comes to the guide RNA, what we see is that the guide RNA will help in making or in introducing this homologous template. Now when it comes to this homologous template, what we see is that homologous templates are like, let's say, the guide. Alright, like the notes that you have. For example, let's say when you're going into the UPSC examination, you have all your class notes with you, and you can see that and you can mark your answers, or you can write your answers in the mains examination. Will it not be easier for you? It will be. So that is what homologous templates are. Homologous templates basically are your revision notes. So all these revision notes is what you are carrying, and that is what is being done here, that there is a guide manual, there is a guidebook that you have as a part of the CRISPR-Cas9 technique, and hence CRISPR-Cas9 technique becomes much more effective because it uses that guide. It just, let's say you are in a new city and you have the map of the city with you, makes it easier for you to travel through the city. So this is what is happening in case of SDN2. So in case of SDN1, there was nothing like that. So you were on your own, you have landed in a new city, you have no map, you have no mobile phone, and that's why doing everything you have to do on your own. You have to talk around, talk to people, understand the city and all that. But in case of SDN2, you have the map with you, and that's why you know how to move throughout the city. So that is what the basic idea is when it comes to SDN2. So in case of SDN1, there is no external guide. In case of SDN2, there is one external guide, which is a guide RNA, which is helping in understanding where do we need to make the editing, where do we need to edit the gene. So that's why SDN2. In case of SDN3, this is where a foreign gene is being introduced. So this is a kind of genetic modification where a gene is being brought in externally. So there is a foreign gene. For example, let's say BT cotton. So in case of BT cotton, you have a foreign gene that is being introduced into the cotton plant. So that's why it is SDN3. It is similar to SDN3. But in case of SDN1 and SDN2, there is no foreign DNA that we are introducing. So that's why this is supposed to be much more safer. Now, why are we discussing these technical terms? Why are we discussing SDN1, 2 and 3 and all that? Understand why. Alright, so according to the new rules that have been brought in by the government of India, they have tried to classify between what is gene editing, that what can be called as gene editing, and what can be called as gene modification. Alright, so gene editing and gene modification. So what is being done here is that if when it comes to gene editing, what the government is saying is that if an editing in the gene has been done and no foreign DNA has been introduced, alright, gene editing is when no foreign DNA is introduced inside the genes, alright, inside the genome. So no foreign DNA introduced, only editing done. Whereas in case of gene modification, they are saying that gene modification would be when you are introducing foreign gene. When we are introducing a foreign gene. Alright, when we are introducing a foreign gene, we'll say that this is gene modification. Now what has been done is that for all these crops where some kind of gene editing or gene modification is being done, what the government of India has said is that if it is using SDN1 and SDN2, this is where you are using SDN1 and 2, alright. So if there is SDN1 and SDN2, this corresponds to gene editing, and if SDN3 is being used, then it corresponds to gene modification. And the rules have been made easier, alright. The rules have been made easier. Getting the go ahead, alright, getting the approval will be easier if it is SDN1 and 2. So here in this case, getting approvals will be easier. Getting the approvals will become easier. So this is what we are seeing here. And in case of SDN3, the rules will remain as they are as of today. So that is what they have said. Alright, this is what they have said when it comes to gene editing versus gene modification, and that's why we are talking about SDN1, SDN2 and SDN3. Alright, so let's go back. So look at the statements given here. These are gene editing techniques used in modern biotechnology for addition, deletion, modification of genes. That's correct. Second, recently the Ministry of Environment, Forest and Climate Change exempted SDN1 and SDN2 categories from concerned biosafety regulations, potentially helping in the faster development of new varieties of crops. This is correct, because now they will not have to go through all the steps which takes a lot of time for any kind of release to happen. So for example, let's say GM mustard we discussed. 2018 it was about to be launched, but the work did not start just couple of years back. The work had started almost 10-12 years back, and since then the work was going on in GM Mustard, and ultimately in 2018 it was being released. That, for example, the productivity tests were carried out in 2011 and 2014, and then after that it was about to be released. So that's why I understand that all these aspects, they become very, very crucial. So both these statements given here are correct, and this is what has been asked of you, that what are the correct statements. So both the statements are correct in this particular case. So this is question number 3. Let's move on. Question number 4, a direct static question. So consider the following statements about Indian Biological Data Center. It is the first national repository for life science and data in India. Second, it is established at the Regional Center of Biotechnology in Faridabad. So these are the two questions given in front of you. These statements, please mark your answer and let's have a discussion on it. Alright, so I see that many of you have marked your answers. So let's have a discussion here. So we are talking about Indian Biological Data Center. Alright, so Indian Biological Data Center is actually the first national repository for life sciences data in India. So for the first time we have a center, we have a national repository which will take care of all the data with regards to life sciences in the country. And especially wherever we are talking about all the data that has been generated from public funded research in India. Alright, for public funded research in India, the government funded researches in India, for all those data that are being generated, all those data will be collected and stored at one repository, and this is what the role of the Indian Biological Data Center is supposed to be. So that's why the first statement is correct. The second statement, it is established at the Regional Center Biotechnology in Faridabad. This is also correct, that this is being done with the help of the National Informatics Center, NIC. With the help of NIC, this is being done, and we basically see that this is how we'll be looking after all the data. We'll be trying to store all the biological data that is being produced in India with regards to the various researches that are happening in the country. So that is basically what is being done. Alright, so now both the statements, statement number 1 and statement number 2, both the statements in this particular case, both are correct. Moving on, question number 5. Consider the following statements about Human Genome Project. First, it is aimed to sequence the entire human genome in addition to the genomes of several carefully selected non-human organisms. This is the first statement. The second statement, the sequence of the human genome generated by the Human Genome Project was not from a single person. So these are the two statements given. So we are launching the poll for you. Please mark your answers. Let's have a discussion here. Well, it's some suggestions flowing here with regards to whether to read class 11th and 12th biology books or not. I would suggest against it, apart from just one unit in your class 12th book that is about environment and ecology, unit number 10, where you have a couple of chapters with regards to environment and ecology. At most you should read that, but don't read anything else. For example, some of you have asked about biotechnology. I would suggest against it. You don't need to read class 12th biology book, NCRT biology book. It could be too technical for you. I would rather suggest that you go through other sources, your lectures and the current affairs. That would be much better for you, I would suggest. So, and especially if you are from non-science background, it will be a very, very difficult task for you to go through class 11th and 12th biology books. So I would not suggest it. Alright, so let's discuss this. Consider the following statements about the Human Genome Project. Now, why are we discussing this Human Genome Project? The reason is that it was completed in 2003, and this year we are in the 20th year of this completion, right? I am still living in 2013, I think. So we are in the 20th year of its completion, and that's the reason why are we talking about this Human Genome Project again. Now, when it comes to the Human Genome Project, it was a project that was taken in early 1990s. I think 1990 was exactly the year when it was taken. And as a part of this Human Genome Project, first we started with other organisms. So it's not that we directly started with humans only. Other organisms also were studied. Their genomic sequences were also studied. And then we came to the human genome. Alright. So first and foremost, when we say human genome, what do we mean by genome? So you have these terms, right? DNA, gene, and then comes the genome. Alright, so when we talk about DNA, DNA is the most basic unit. These are the most basic proteins which help in carrying all the genetic information inside an organism. Any organism, whether plant, whether animal, whether humans, any organism. And genes basically are sequences of DNA. Alright, these are sequences of DNA. So bunch of DNA together will make up a gene. And this entire set of gene will be called as the genome. Alright, so this is basically what we say. So for example, let's say letters in the English language A, B, C, D would be the DNA. The words, when you make meaningful words out of it, they are called as genes. And the entire language that is English will be called as genome. In between, there is another term. Some of you, I'm sure you will ask, what do we mean by chromosome? So chromosome, you can say that these are sentences that you're forming out of these words. Alright, so DNA basically are the letters, the genes are the words, the chromosomes are the sentences that you are forming out of them, and the entire language system that you have is the genome. So when we say the Human Genome Project, we are talking about the entire human genome, the entire genetic sequence that humans have. So completes 20 years. Some of the discussions have been happening that there are still some gaps that we have here, and we still haven't been able to completely map the entire human genome. We have not been able to completely exactly sequence it up, but they are saying that maybe this is the year when we'll be able to do that finally, because there were some of the missing sequences that also have been mapped in the last couple of years. So, aimed at sequencing the entire human genome in addition to the genomes of several carefully selected non-human organisms. This is correct. That there were other organisms also which were a part of this particular mission. Then after that, the second statement says, the sequence of the human genome generated by the Human Genome Project was not from a single person. This is also correct, that it was not from a single person, because every person will have a different set of, there will be some different sequences here and there, and there are a lot of repetitive sequences. The basic problem that we have been facing in doing the complete sequencing is because of these repetitive sequences that we have in our body. And these repetitive sequences, just know the word, no need to go into the details of it. They are also called as satellite DNA. So these satellite DNA, they basically are related to the repetitive sequences that we have in the body. Alright, the repetitive sequences that we have in our body, they are called as satellite DNA, and they are the biggest culprits why we are so why it makes it so difficult for the scientists to map the entire set of genomes, because the satellite DNA will also be different in various different persons. So that's why taking only a single person would not have been enough. So that's why there were a lot of volunteers who were also participants in this human genome project, and all their DNA was taken, and then the mapping of the entire human genome was being tried. So that's why both these statements that have been given here, both the statements are correct. So correct answer here will be C, that is both 1 and 2. Moving on, question number 6. A 2 million year old ecosystem has been identified as world's oldest DNA. This was recently discovered where? Alright, Iceland, Greenland, Africa, or Siberia. So please mark your answers. Let's have a discussion here. Alright, so most of you have marked your answers. So let's have a discussion here. Alright, so we are talking about a 2 million years old ecosystem that has been identified as having the oldest DNA. I mean, it has the oldest DNA. So what exactly are we talking about? First of all, the answer is Greenland, but more than the answer being Greenland, it's about the topic. Now, when we are talking about this oldest DNA, until now the oldest DNA that we had a record from Siberia. Alright, so from Siberia, the DNA records that we had were almost 1.2 million years old. Alright, 1.2 million years old. Now, in this scenario, when it comes to what we have discovered in Iceland, we see that this now goes to almost 2 million years back, which is extraordinary. Now, what they have found, basically they have found some sequences which show them that there are perhaps possibilities of horseshoe crab being there, or even mastodons being there, or even reindeers also, possibility of reindeers also being there. So all these sequences have been found. Now the sequences that they have found, basically they are related to this ecosystem. Now there is something, again, another kind of DNA. We talked about satellite DNA in the last question. There is another DNA called as eDNA. Now when we say eDNA, it has nothing to do with websites, or it has nothing to do with the internet. But when it comes to eDNA basically is something that we call as environmental DNA. Environmental DNA. Alright. So when it comes to environmental DNA, these are DNA sequences that we find from some given ecosystem. Let's say you are analyzing soil and you are deriving some DNA from the soil samples, then it is called as environmental DNA. If it is being derived from, let's say, any organism, then it's the normal DNA sequence. But let's say you have not derived it from any animal or any plant, but rather you have picked it from the soil, then in that case it will be called as environmental DNA. Alright, so this is what they have done, that they had taken some environmental DNA, and from these environmental DNA is where from they actually have gotten the sequence. And this is where they got the hint that until this, actually the DNA samples that they have found in Greenland, this is the oldest DNA sample that they have found until now, and that's why it is an extraordinary sequence here. Now also understand that why Greenland, and before that also why Siberia. What is common between Greenland and Siberia? the commonality here is that both of these areas, they are cold areas, right? These are frozen areas. So in these frozen areas, today they are frozen, but they were not always frozen, right? So for example, for Greenland also, it is said that the temperature at the time might have been at least 20 degrees higher than the temperatures that you see today in Greenland. So that's why the temperature ranges were different, the climatic conditions were different. They may have been more tropical, more temperate in their nature compared to what they are today. So that's why there might have been some of these organisms that thrived at that point of time, but then after that, once they have become cold, and now that there are layers of ice that you have in these kind of areas, that also prohibits you from understanding or having those DNA samples in front of you. And also the fact that because of it, it can also survive for a longer duration. That also becomes a reason why it would be able to survive for a longer duration as compared to if it was in the tropical region where there was no ice or there was nothing that would preserve the DNA for such a long time. So that's why understand why these cold areas are often the areas from where we get these DNA samples. Alright, moving on. The next question. Consider the following statements about HTBT cotton. HTBT cotton is resistant to the herbicide glyphosate. Second, its illegal cultivation has increased in the last couple of years. These are the two statements given, and please mark your answers here first. Yeah, the permafrost area, as many of you have pointed out, that's correct. All right. Yes, Benazir, you are correct that we have been asked about that which of the following statements are incorrect. Many of you, I think, did not see that. So here we are talking about these two statements. So let's start the discussion here. So we are talking about HTBT cotton. All right, HTBT cotton. So first and foremost, what do we mean by HTBT cotton? So when we say BT cotton, we know that BT cotton, what does it do? BT cotton will be able to help the cotton plants or cotton crops against bollworm, against a kind of pest. When we say HT, any crop that is an HT variant, HT means what? HT means herbicide tolerant. Herbicide tolerant, meaning that they would be able to tolerate the herbicides. What do we mean by herbicides? Herbicides are supposed to be used against any kind of weed that is growing in the given crop field. So any unwanted plant that is growing, any weed that is growing, against them we use the herbicides. So herbicides will just kill these weeds. And at the same time, we also want to protect our crops. So to protect these crops, we need the herbicide tolerant crops or herbicide tolerant varieties, so that even if you are using the herbicides, it will have no negative impact on the crops that are being grown. This is what HT variant is. So HTBT cotton is something that has been in the news, mainly because of wrong reasons, mainly because of the fact that we have seen that the illegal cultivation has actually increased. We have seen that there have been a higher number of cases where illegal cultivation of this HTBT cotton is being done, because ultimately when it comes to the Federation of Seed Industry of India, FSSI, so they have asked the government of India to help them go forward with the release of these seeds, and then they want to release the seeds for the Indian markets, for the Indian farmers. But what we have seen is that it is already being cultivated illegally, and in some parts of the country in very large amounts. That is that actually they are saying that up to 10 to 15% of all the cotton that is being grown perhaps is already HTBT cotton. So that's why the illegal cultivation has grown in some of the states, especially like in Maharashtra, in Gujarat, in parts of Andhra Pradesh, Telangana as well. We see that in all these areas, we have seen that this is already being grown illegally. So that's why the second statement is correct. The first statement says HTBT cotton is resistant to the herbicide, that is glyphosate, and this is also correct, and that's why herbicide tolerant, that it will be able to tolerate glyphosate. So that is basically what we are trying to. Both the statements given here are correct, but you have been asked what is incorrect. So that's why the correct answer here will be what? Sorry. The correct answer here will be D. That is neither 1 nor 2. Right? So please always remember, always when we say read the questions carefully, this is what we mean by read the questions carefully. But we know that many of you might go wrong because of this very statement. Moving on, question number 8. So for the first 7 questions, we have discussed some of the key areas with regards to biotechnology. Now in the second segment, we move towards some of the questions which are pertaining to IT and communication. So let's have discussion here. Consider the following statements regarding 5G network. 1. Maximum internet data speed is 50 Gbps. It does not support 3D virtual reality. It offers high latency. Which of the above statements are incorrect? And again, 4 options have been given to you. Please mark your answers and let's have a discussion on it. All right. So I think most of you have marked it. So let's start the discussion here. So we are talking about 5G network, and 5G network, again, more than the question that we are discussing, the topic. The topic perhaps is one of the most important topics that you are going with in case of science and technology this year. So 5G network becomes crucially important. So let's start the discussion. First, maximum internet data speed is 50 Gbps. No, it is not 50 Gbps. Maximum that we might see here might be up to 10 Gbps. Maximum, some report that they might do some improvements, that it might move up to 20, but even 20 will not happen. In most of the cases we'll see that 10 will be the ceiling. So that's why in this case this is wrong, that maximum data speed will not go up to 50 Gbps. It will perhaps go up to 10 Gbps maximum. Maybe they will be able to improve upon it somehow, but not beyond maximum in this case, not beyond this 10 Gbps limit. Second, it does not support virtual reality. This is wrong, because when it comes to virtual reality, 3D virtual reality, already 4G is supporting it. 4G has already started supporting it, so definitely 5G is also going to support it. So that's why saying this, that it will not support virtual reality, forget about just virtual reality. Any advanced concept, any advanced technology that you know of, all these advanced technologies, they will get a lot of help from 5G. And that is why today we are saying that 5G is where the mobile phone network now moves beyond just your personal use. Because until now, until 4G, you are using mobile phone networks for your own purpose. Maybe your official work, maybe watching a lecture maybe on this network, or maybe using it for your college work, maybe using it for various different purposes. So, but essentially what we see is that the purposes are personal in nature right now. Correct? Most of these are personal in nature right now. So first and foremost, when it comes to 5G, we say that now we are going to move beyond the personal use. So why we are saying that it moves beyond the personal use? Because for the first time, we might start using it in various different sectors. For example, the healthcare sector might start using 5G. The agricultural sector might get a lot of help from 5G. So we might see that all these sectors might start getting some help from 5G network because of the speed that you're getting, the kind of network connectivity that you're getting with 5G, and not only network connectivity, the kind of latency, the kind of low latency that it offers. So this is also wrong. Alright, it offers very low to almost zero latency, you can say, offers almost zero latency. So the latency can be as low as just 1 millisecond. Latency can be as low as just 1 millisecond. So what do we mean by latency? Latency. So when it comes to the term latency, latency is nothing but the delay. In simple terms, you can say the delay or the lag that you have in the network. The delay or the lag in the network is called as latency. So when we say delay in the network or lag in the network, many times you would have faced this in one form or the other. For example, let's say if you're watching a video, you sometimes might see that the voice is coming first and the video later, or maybe perhaps the pictures first and the voice later. So that happens because these are two different waveforms. These are two different waves and these are traveling independently. They are synchronized and they are made to travel. But it may so happen because of some kind of problem, maybe with a problem with the device, that the device is not good enough, maybe because of the network, that the network itself is not good enough, maybe because of the internet speed. There can be multiple different factors, and one of these factors might lead to one moving in the at the normal rate and the second falling behind. So you might have seen this. So these kind of delays that can happen in the network, any kind of delay in the network will be called as latency. So with 5G network, we are able to reduce that latency. We are able to reduce that latency even further. And that is what we are looking at when it comes to 5G network, that it will provide low latency altogether. So that's why high latency is wrong. So you have been asked what is incorrect. So all the three statements are incorrect. So correct answer, that's why here will be D. So remember that when it comes to 5G as a topic will remain very, very important, especially when it comes to this year's examination. Now, although most of you had marked D here, something that I often keep saying, that many a times when all the statements are wrong, it plays with our confidence. So please make sure that it doesn't happen with you when you see these questions. Always go statement by statement. If you have any problem with any statement, go about try to understand that and eliminate it. So 5G as a topic will be very, very important. Then after that, there is this 5GI, which will be very, very important, because 5GI is being considered in the context of India, that there is a modified version of 5G that India wants to use, which has been named as 5GI. And then again, there is a document that has been released for 6G also, that how exactly India will move towards 6G by 2030. So there is a document, there is a project document that has already been released. So basically you can say that in a manner, India wants to have the first move advantage, that we want to be the first one to reach the next generation of mobile networks. So that's why already we see that there is a document that has been released by the government of India, so that they can start working towards a plan for introducing 6G network in India by the year 2030. So this is something, that's why all these topics related to this, all the properties of 5G, everything will be very, very important. So please consider all these things before going to your examination. Moving on, question number 9. Consider the following statements with respect to quantum technology. First, it is the research at the subatomic scale of matter and energy. Second, it may help us understand avian navigation. Third, China is the only country to be working on advanced quantum technologies. 3 statements in front of you. I would say it's an easy question, but please mark your answers and let's have a discussion on it. Please compare with 6G document. I would say not really required. It's just that we are looking at improvements everywhere, that we want to improve the speed further, we want to improve upon the latency factor. So there are a lot of technicalities that have been discussed there, which I suppose is not really important directly. Alright, so it becomes an easy question, I know, and I see that most of you have marked the correct answer. Alright, so let's understand what question number 9 says. Let's start the discussion here. So we are talking about quantum technologies, and in case of quantum technologies, the first statement is it is the research at the subatomic scale of matter and energy. This is correct, that when we say quantum, when we are talking about quantum mechanics or quantum physics, we basically are talking about what? We are talking about the subatomic particles and the properties that we see in the subatomic particles, their energy calculations, their movements, and each and every property that they have. When we do the calculations there, when we do the computations there, when we are looking and trying to understand the properties of these particles, this is what we call as quantum mechanics or quantum physics, and hence the technology that is associated to all these subatomic particles and their properties, this is called as quantum technology. Now, when it comes to quantum technology, understand this, that when it comes to quantum technology, in case of quantum technology, what we see is that the properties at this level are very different compared to what we see at our level. Compared to that, the properties that you see at this level is very different, and that's why the properties for us might also seem very weird, but they do happen, they do exist, and that's why we see that it has a very important role to play for all of us. So first and foremost, we are talking about the research at the subatomic scale of matter and energy. This is perfectly correct. Second statement, it may help us understand avian navigation. This is actually correct. Now understand this, that when it comes to quantum technologies or any new technology that we are discussing, in most of these kind of questions where you have that whether this new technology can do this or do that, or whether it can do this or do that or not, in most of the cases it will be correct that whatever application they have talked about, that is an application of that technology, unless it's a very extreme case, unless it's a very extreme technology, that's something that they are talking about. So unless it's a very extreme thing, you can consider that most of the normal application areas, if they are talking about, they must be correct. And that is what we see here also, that it will help in avian navigation. Not only avian navigation, there are so many things in biology, there are so many things in genetics, there are so many things in chemistry, there are so many things in physics, there are so many ways in which we see that quantum technologies will help. There are many different sectors where quantum technologies would help. So that's why quantum technologies, once they are adopted in its full reality, at its full scale, we'll see that it will completely once again change the world, as we say. So that's why all these application areas, you can consider that they are going to be very much doable with regards to quantum technology. So yes, this is also correct. Third statement, China is the only country. Now first of all, only, an extreme word, most of the times it's wrong, to be working on advanced quantum technology. It would be wrong to say so, because there are so many countries around the world that have been working on quantum technologies. But there is one important thing about China that you must understand when it comes to quantum technologies. I think there is no other country that has made so much of advancement in terms of quantum technologies as much as China has done. So if it is about whether China is the leading country, I would say yes. Compared to Europe, compared to the US, China has gone far beyond whatever has been achieved until now by Europe or in US or other parts of the world. So yes, China is the leading nation. And that's why if you look at the total investment that has been done in terms of quantum technology, as per the data of 2022, a total of almost $15 billion is what they are saying already has been invested by China in terms of quantum technologies. Compared to that, when you look at other countries, you'll find that other countries are falling way behind. Other countries are falling way behind, that even when it comes to US, for example, US has done an investment of almost $1.5 billion. Compared to that, China, we are talking about $15 billion. So that's why China already has been moving very fast when it comes to quantum technologies. So is it the leading nation? Yes, it is. Is it the nation with the highest amount of investment in quantum technologies? Yes, it is. But is it the only country working on advanced quantum technologies? No, it's not. There are other countries, and there are so many different countries which are working on it, whether it is US, whether it is about the other European countries like France, like Germany, like the UK. There are so many countries who are working on it. Russia has been working on it. Japan is working. South Korea is working. India, of course, is working. Australia is working. So there are so many other countries which are also working on these advanced technologies. But yes, understand this, that when it comes to how much of advancements have been made, there China is right now at the top. So statement number 3 is wrong, and you have statement number 3 in 3 of the 4 statements. So anyway, the correct answer here will be A. Alright. So that's why when it comes to quantum technologies, there are, as I told you, that there are multiple different sectors where quantum technologies will start helping. And that's why we'll see that very soon quantum technologies, we already see that India is also investing, and very soon we'll see that other countries will also start investing, and they'll also be trying to get to understand how to use quantum technologies as quickly as possible. Moving on. Question number 10. 4D printing has applications in which of the following? Uniform of a soldier, home appliances, footwear of children, and jewelry. Alright, so 4 statements given in front of you. If the fourth one is, if option D is not available, not visible, just in case, it's again all of the above. D is all of the above. Computing, simulation, communication, and sensing and metrology are the 4 pillars of quantum technologies. Absolutely correct. That these are 4 basic areas where we have been working. So yes, you are right there. So most of you have marked it. Nice explanation in Hindi, sir. Oh yeah, so Hindi class is going on in the morning and English going on in the evening. For me also it becomes difficult at times because I've completely have to switch from a complete English to complete Hindi again. So not a very easy task altogether. Alright, so let's come back. So question number 10, talking about 4D printing. So 4D printing, when it comes to 4D printing, understand what is 4D printing. So until now we have been talking a lot about 3D printing. For the last 10-15 years we have been in 3D printing, and that's why 3D printing is being discussed a lot. Now we come to the era of 4D printing. Now what is this 4D printing? How is it different from 3D printing? So until now, with regards to when it comes to 3D printing, we see that this is where the changes in shapes and changes in forms was not easy when it comes to 3D. Only whatever material is being reproduced, whatever material you want to produce, that can be done with the help of 3D printing technology. But in case of 4D printing, now we move one step ahead. And in this 4D printing, that's why now we are saying that we will be able to change the forms with regards to some of the external external factors as well. So for example, let's say when it comes to external energy being provided, so externally heat being provided, 4D printing can respond to all of that. 4D printing can respond to changing forms. So that's why when it comes to clothing, it can become a very, very crucial form in the use when it comes to clothing. So that's why when it comes to, let's say, footwear, or whether it comes to uniforms, in all these cases we can make the use of 4D printing. So in that's why just remember basics, that when it comes to 4D printing, we are saying that 4D printing is where they'll be able to change the forms, that they'll be able to change themselves with regards to the changing forms, and that is what the basic idea behind 4D printing is, and this is where the beauty of 4D printing comes in. So again, coming back to what all can be done, as I told you that any new technology, any new advanced technology that they are talking about, most of the cases you will find that they can be used in all these application areas. Alright, so that's why uniform of a soldier, yes, doable. Home appliances, yes, doable. Footwear of children and jewelry, sorry, the spelling of jewelry is wrong here. So all these four aspects, we see that they can be used. So that's why the correct answer will be all of the above, that it can be used in all these cases. So just remember that when it comes to 4D printing, what is different? How is it different from 3D printing? The basic difference is that now, according to the changing external environment, according to the changing, you can say, changing energy or change in heat or any kind of factors, we see that 4D printing will be able to respond to it, and hence it will be useful in all these situations. Moving on, question number 11. Consider the following pairs, terms sometimes seen in the news, the context or the topic in which they are being used. First, cryptojacking, second, gigamesh, and third is YOTA. Which of the following cases, which of the following given are correctly matched? Please mark your answers quickly. Let's then start discussing it. All right, so let's start discussing it. So first is cryptojacking with regards to cyber attack. Is this correctly matched? Yes, this is correctly matched. So in case of cryptojacking, when it comes to cryptojacking, what is cryptojacking? Just understand this, that cryptojacking is when, so another term, let's start with another term, that is crypto mining. Crypto mining. So first and foremost, crypto mining. What is crypto mining? Crypto mining, in a manner you can say, is creation of new cryptocurrencies. All right, so let's say in case of bitcoins, so creation of bitcoins or new bitcoins would be called as crypto mining. So these crypto mining will be done by users who are there in the network called as nodes. So these nodes will do the mining. They're also called as miners. So they will do the mining and they would basically create new bitcoins or create new cryptocurrencies. So that is basically what is happening here. Now, crypto mining, sometimes what may happen is that, say, let's say you are doing the crypto mining, but I have already hacked into your system. So whatever new cryptocurrencies you are creating, actually it's not going to you, it's something that is being hacked by me, and that's why it comes to me, and ultimately I become the miner. I would be the apparent miner of this cryptocurrency, and that's why all the advantages, all the financial benefits that you might have drawn, you would not draw it, but I would draw it. So this phenomena is called as cryptojacking, where the crypto miner has been hacked, or the system used by the crypto miner has been hacked, and because of that, whatever new cryptocurrencies they are creating, whatever new cryptocurrencies they are producing, they are not getting any benefit out of it. All the benefits are coming to the jacker, the crypto jacker. So that is what crypto jacking is. So yes, this is a kind of a cyber attack where there is some kind of malware that has been introduced into the crypto miners system so that they can actually use it against them. Then after that, the second one is gigamesh. Now, is gigamesh related to cryptocurrency? No, it is not. Gigamesh, when it comes to gigamesh basically is a kind of a network technology that helps 5G. All right, it is something that helps 5G in something which is called as backhaul, or in creating backhaul capacity. Creating the backhaul capacity. Backhaul capacity basically is where you are creating some kind of connection between the networks with the help of generally wires. That generally fiber optic cables will be used to connect one set of network to another network, or a sub network to another network. So this is something which is called as backhaul, and this backhaul is done with the help of fiber optic cables. But here in case of 5G, we are trying to do this backhaul wirelessly. So that is what giga mesh technology is about, that you are trying to do this black backhaul, or trying to produce connections between networks or sub network categories as without using the cables, but instead of doing it wirelessly. So that's why we see that it is not related to cryptocurrencies. And then after that you have IOTA D1. Now IOTA D1, basically just understand this, that IOTA D1 is nothing but a data center. All right, this is a data center, but the country's largest data center. This is going to be the country's largest data center. Almost 5,000 crore rupees have been spent on making this data center, and this becomes the country's largest data center. So again, it is not related to 5G at all. So what is correctly matched has been asked of you. So the correct answer here will be one only. One only, that's the only statement that has been correctly matched. The second and the third statements are not correctly matched. All right. Moving on, question number 12. What is or what are the features of Web 5.0? It allows the users to own their identity on the internet. It maps the individual's feelings in real time. It is a mix of Web 2.0 and 3.0, and then all of the above. So please mark your answers. Let's have a discussion here. All right, so let's start the discussion here. So we are talking about Web 5.0, or as some of you might call as Web 5.0. So whether Web 5.0 or Web 5.0, what we are basically talking about is a new generation of the internet. All right, a new generation of the internet. Now when it comes to new generation of the internet, what we are saying is that this is a new generation, Web 5.0. Why are we saying it is 5.0? Because it is the fifth generation. So what do we mean by fifth generation? There must have been 4 other generations then. So starting, for example, when you say Web 1.0, so then in case of 1.0, when we say that, that's the basic use of internet. When the internet started, that basic websites could be created. So these basic websites, they can just display the information and you can just read the information. So read only applications is what we had in the first era. Then the second generation, Web 2.0. So in case of 2.0, we see that this is where you can also interact with the internet. For example, Facebook, Orkut. If you remember, if you know what is Orkut. So Facebook, Twitter, all these basically will be taken as examples of 2.0. All right, and then after that comes Web 3.0. And when it comes to Web 3.0, this is where we are saying that more of automation is coming, more of data analytics is coming into the picture. So all these basically will be said to be a part of Web 3.0. All right, and then after that, when it comes to Web 4.0, in case of Web 4.0, we see that this is where we start also going towards virtual reality, creating virtual reality spaces, etc. So that becomes 4.0. And then comes 5.0. Now in case of 5.0, when we say it is Web 5.0, this is where first of all we are talking about a platform which will have some newer aspects being introduced into the web platforms. So when it comes to the web platforms, so first and foremost, does it allow the users to own their identity on the internet? Yes, it does. So there are aspects where we see that it will help the owner, or it will help the user to own their identity, and according to their identity they can basically be there, have their presence online. Maps individuals' feelings in real time. This is also correct. What we are saying about Web 5.0 is that it will also bring in a factor of emotional intelligence. It will bring in a factor of emotional intelligence as well in case of uh, the feelings, and that's why we are talking about feelings also being a part of it. So when we say feeling, it will also use a lot of softwares, for example, lot of methods, technologies, for example, even facial recognition. So when it comes to facial recognition, it might use facial recognition to understand what is happening and what kind of user do we have. For example, let's say you are somebody who likes using headphones all the time. So that can be mapped, and accordingly when the content is being given, whenever any product or any, let's say, audio or music or any video is being given to you, it will be also optimized in a manner that somebody who uses these headphones, it should be optimized for them. So that's why we see that we are talking about feelings, we are talking about your state, we are talking about everything externally, so that you can own your internet, you can use the internet according to your requirements. So that is also correct. Mix of web 2.0 and 3.0, yes, we can say that it is basically carrying all the aspects of 2.0 and 3.0 together. So that's why the correct answer will be all of the above. That all these statements given above, they are all correct in the context of this particular question. So that's why the correct answer here will be D, that is all of the above. So web 5.0, again, because now being introduced, can be important from examination perspective. Now coming to the next question, what are the applications of metaverse? Merged offline and online commerce, virtual reality 3D clinics, 3D avatars of students and teachers, and virtual art galleries. So these are the 4 statements given, 4 applications given in front of you. Please mark your answers. Let's have a discussion on it. Alright, so here I see that many of you have marked your answers. So audience poll is correct here, just like in KBC. So it's correct that most of you have given the right answer. Now coming to what is metaverse. Now there are so many technologies that we are talking about. That metaverse, all of a sudden in 2022 there were so many discussions about it. Just before that, NFT was one term that so many of us were discussing about. But we have forgotten about all that, and ChatGPT is right now the technology that everybody is discussing. So first and foremost, let's try to understand first that what do we mean by metaverse. So metaverse, in a manner, you can say that it is nothing but a digital extension of the physical reality. All right. So this is what we are trying to do, that we are just creating a digital extension of our physical reality, and that's why in a manner you can say that it combines the technologies of virtual reality and augmented reality to make this happen. All right, so that's why we are saying that this is nothing but a digital extension. In simple terms, we'll say that it is a digital extension of our physical reality. Now what do we mean? What is the meaning of this phrase, that this is a digital extension of our physical reality? Now understand this, that when it comes to our physical reality, if we can create a digital space which is perfectly matching with our physical reality, it will become our metaverse. So for example, let's say your home. If you can create a digital version of your home, each and every room and each and everything kept in those rooms, if you can just create a virtual an online version of it, then in that case we'll say that this is what will be called as virtual reality, or this will be called as your metaverse. Now this metaverse becomes very useful, and all of a sudden last couple of years, especially 2022, we see that this was something that was very highly being discussed all across the world. And Facebook basically changed its name, also became Meta, as the name of the company itself changed. So now what we see is that in this particular case, what happens is that it can have a lot of applications. That, for example, if it has to be used in commerce, how exactly? Let's say I am a simple shopkeeper. I have a simple grocery store. Now what can I do? I can create a metaverse of my grocery store. And this metaverse will be available for everyone. You also, let's say, are there. You also have an online presence. You also have a presence in this metaverse. So instead of you, let's say, from you, my grocery store perhaps is 10 kilometers away. So you don't have to now travel 10 kilometers. You don't even have to order online. You can simply go into the metaverse, and there you can physically just go and pick whatever you want. Pick all the grocery items that you want, and the billing will be done. And then the product will not be delivered online. The product will not be delivered as just a virtual reality, but actual products can be delivered to you. So in this manner, it can be used in commerce, that an online extension of the offline commerce can be created. Similarly in education, let's suppose right now I am in my physical form here. You are all, I can just see you in your comments and nothing else. So let's say we can create a virtual reality classroom. We can create a metaverse of this classroom where I am existing in my avatar in whatever form I want. Maybe I can just exist there as Batman if I want. And you all also can create your own metaverse. You can also create your own avatars, and you can also be there in that virtual classroom. And then it will be a much better interaction perhaps. Even though we are interacting online, it can be a much better interaction. So that's why we see that wherever we are talking about this digital extension of our physical reality, this is what metaverse is. So let's go back. What are we talking about? So merged offline and online commerce, yes, possible. Virtual 3D clinics, yes, possible. 3D avatars of students and teachers, yes, possible. Virtual art galleries, yes, of course, possible. So all the four are possibilities. So you have been asked to mark the one that is correct. So all the four given here, all the four are correct. So question number 13, answer will be B. Now moving on to the last segment of our discussion today, and this is about the Nobel Prizes. So here we have last 6 questions, and these 6 questions are about the Nobel Prize of last 2 years. Nobel Prize Physics, Chemistry and Medicine of 2021 and Nobel Prize of Physics, Chemistry and Medicine 2022. So the first question, question number 14. Consider the following statements concerning paleogenomics. It is the study of present-day genomics to predict the genetic makeup of our ancestors. The 2022 Nobel Prize in Physiology was awarded for research in paleogenomics, and paleogenomics can help in a better understanding of human evolution. So these are the 3 statements given. Please mark your answers. Let's have a discussion here. Alright, sorry, here also D is missing. So D is all. Here also it is all of the above. Alright, so let's discuss this. So we are talking about paleogenomics. Now when it comes to paleogenomics, paleo. So when it comes to this term is often related to anything that is ancient. Anything that is ancient, study of anything that is ancient, we'll say that it is paleogenomics. All right. So now study of present day genomics to predict the genetic makeup of our ancestors. Not the study of present day genomics. It is the study of the ancient genes. All right. It is the study of ancient genes. So we are studying the ancient genes and trying to understand how evolution happened, how we came from various organisms, how do we share so many genes with so many other organisms. So when we are talking about all these things, this is what paleogenomics is all about. So that's why when it comes to this actually is wrong. All right, the first statement is not correct. The first statement that talks about present day genomics, no, it's not about studying the present day genomics, but rather it is about studying the ancient genes and trying to link them with the present genes. This is what we are trying to do here. So that's why this is wrong. Now, first is wrong. Second is 2022 Nobel Prize in Physiology was awarded for research in paleogenomics. This is correct. Can help in better understanding of human evolution. This is correct. That once we understand the ancient genes, once we understand the connection between the modern genes, or rather the present day genes and the ancient genes, it might help us understand better the evolution that has happened in the humans. So that's why in this case 2 and 3 are correct. So the correct answer here will be C, that is 2 and 3 only. Moving on, question number 15. Consider the following statements concerning click chemistry. They are a set of reactions that are modular and can be easily combined to generate complex molecules. One of the advantages of click chemistry is that it uses a small number of starting materials and reagents and generates minimal waste. So these are two statements given. Again, sorry, D is missing here as well. So D again is neither. D here is neither. Neither 1 nor 2. All right. All right, so let's start our discussion. So now, first and foremost, when it comes to click chemistry, understand what is click chemistry. Click is not the click of the mouse here, but rather this click is the click between two components. So you might have used these kind of bags. I mean, today I don't know, I don't see these kind of bags a lot, but there used to be these bags where you have these kind of buckles that will just simply fit into one another. So this is basically what we are saying, that click chemistry is when there are two molecules, there are two components of molecules that can set very easily in one another, that can fit into one another. Alright, so now what happens is that when you have these kind of components, in these kind of components, what happens is that overall carrying out this process is much simpler, and not only simpler but also overall wastage of the product, the use of the various reagents, etc. is much lesser. So that's why it also becomes cost effective, also becomes one of the key areas in which biochemistry can actually see a lot of advancement. So when it comes to, I mean, I'll not again go into the details of it because the details of click chemistry is very technical in nature, but just understand this, that when we say click chemistry, why we are saying click chemistry is mainly because these are two molecules that can fit into one another, just like what we see in these kind of buckles. So that is what is happening here, and hence we are talking about, hence we are calling it as click chemistry. So in case of click chemistry, this is what we are talking about it. And then again there is another term along with click chemistry that is always in news, is bioorthogonal. Bioorthogonal chemistry. Bioorthogonal chemistry. So where we are talking about the reactions that can happen in some of the living organisms, where in any given living system or any biomolecule for that matter, without having any kind of interference with all the native biomolecules, which is also a part of, which also you can say in a manner it derives itself from click chemistry. So both of these click chemistry and bioorthogonal chemistry, these are very new branches of chemistry which were introduced by the scientists who have been given the Nobel Prize for introducing click chemistry. So set of reactions that are modular, correct, can be easily combined to generate complex molecules. That is correct, and that's why making these complex molecules out of simpler chemical molecules makes it easier with the help of click chemistry. So first is correct. One of the advantages is that uses small number of starting materials. This is also correct, and reagents and generates minimal waste. This is also correct. That overall the entire procedure, which is otherwise going to be very difficult, complex, and very time taking, all of that is reduced. The waste is also reduced. The number of reagents, the catalysts, etc. being used is all reduced. So that is what we see. So both the statements given here are actually correct. So correct answer will be C. Moving on, question number 16. Consider the following statements with regards to 2022 Nobel Prize winners in physics. John Clauser developed an experimental setup capable of producing pairs of entangled photons. Anton Zeilinger developed an experimental setup that addresses a critical loophole in earlier experiments. An alien aspect demonstrated quantum teleportation. So these are the three statements given in front of you. This perhaps is the most difficult question that I have given you in combined today's and yesterday's class, I feel. But if you know, let me know. If you don't, let's have a discussion. No problem. Alright, so let's discuss this. So I know, these kind of questions, if they come, they can be very difficult for you, and most of the times I would say that these are questions that should be skipped, because if you don't know, because name based questions first of all are not supposed to come, but if they do, if you know the names, great, but if you don't, then in that case it becomes impossible for you to mark what is correct and what is wrong. So here statement number 2 and statement number 3, they are interchanged. That what has been written for statement 2 is correct for the statement 3, for the scientist of statement number 3, and what has been written for statement number 3 is correct for statement 2. So only one is correct. I knew that in these kind of questions most of you will mark D, but yes, in these kind of questions perhaps refrain from marking when it comes to the actual examination. Here it's fine, you can mark as much as you want, but yes, remembering the names, I'm just telling you that I don't feel that remembering the names are important. The names can become only important when they have done something extraordinary, something which is first of its kind. Then in that case perhaps names are more important. Otherwise I would say that the names are not that important. But for example, okay, let me just go back to the first question here, the previous question, when we are talking about click chemistry. Name in this case of click chemistry perhaps will be more important, because this is a new branch that has been developed by the scientist who got the Nobel Prize. The name is Barry Sharpless. So maybe his name is more important, maybe here the names are not important, but still I just wanted to give you a question like this. But overall just understand that when it comes to the Nobel Prize of 2022, it was related to quantum entanglement. It was related to quantum entanglement, and when it comes to quantum entanglement, just understand this, that quantum entanglement, what exactly is it? Where we are talking about just two particles, and there are these two subatomic particles whose properties are connected, right? Whose properties are connected in a manner, that if you do something to one particle, if you do any kind of modification, perhaps let's say to one particle, the second will also show the same. Whatever changes you try in the first particle, the second particle will also show the same, if their properties are connected. This is called as quantum entanglement. So in case of quantum entanglement, it is something that has been seen in some of the quantum particles, in electrons, in photons. We have seen that they show quantum entanglement, that if their properties are connected, then in that case what we see is that if their properties are connected, even if they are separated by a distance, even then they would show the same properties, which is very unique, very even difficult to even think of that something like this is even happening. When Albert Einstein discovered it for the first time, he named it as spooky action at distance, that some spooky action is happening at a distance, and hence he named it as spooky action at distance. So that is what quantum entanglement is. So in case of quantum entanglement, or let's say quantum teleportation, so quantum teleportation is when we are able to, let's say, when we have these entangled particles, then in that case, if we are able to teleport their properties or demonstrate that their properties being teleported, then in that case it is called as quantum teleportation. All right. So that is basically what we see in particular case. Now overall, when it comes to these concepts, just understand that quantum entanglement or quantum properties in general will be very, very important. So any quantum property, this year's examination is going to be important. Moving on. Question number 17. Consider the following statements with respect to the 2021 Nobel Prize in Physics. It was given for important contributions to our understanding of complex physical systems. It will help in predicting climate change. These are the two statements given. Please mark your answer. Let's have a discussion on it. What shall be the strategy at this point for the prelims examination? You are in the last month, so it's only and only about revision. It's only and only about revising all the topics that you have covered. Don't go through any new topic. Everything, every current affairs aspect, all the things that we have taught you, all the things that you have studied, just go through that. Just revise that as much as possible. Prelims test series, all those things, I mean all the questions covered there, they all can be taken as a reference source so that a good revision can be done. So it's all about revision, about each and every subject that you know, and try to focus more on your strength than on your weaknesses right now. Alright. So question number 17. So question number 17, first of all, it is talking about Nobel Prize in Physics 2021, which was about climate modeling and basic understanding of how climate change, how there are various factors that contribute to climate change. So that is what they were given the Nobel Prize for. These scientists, they discovered, or they basically made a model, they made a mathematical model to understand how climate change happens, how the various aspects of climate change takes place. So that is what they had done. So even the smallest of the things that you know today, without even thinking that we know about it. So for example, let's say in environment and ecology, you have studied about, let's say, the contribution of the greenhouse gases, that what the contribution of carbon dioxide is towards greenhouse effect or towards global climate change. So you have studied this, they studied it for the first time. They were the ones to discover these things. So that's why climate modeling, how exactly climate works, how exactly these greenhouse gases also contribute towards climate change, all these things were done for the first time by these two scientists. So that's why let's go back to the statements given for important contributions to understanding the complex physical systems. This is correct, that when it comes to climate change, I'm sure that in environment and ecology classes you might have come across a lot of different factors that lead to climate change, that contribute to climate change, but how they interact as a very complex system is very, very difficult to comprehend. And that's why we see that there are so many debates, there are so many discussions, there are so many alternate theories about climate change and how climate change is happening and what are the factors that are contributing to climate change. So all this is a very complex geographical system, climatic system that contributes to climate change. So for the first time, these were the scientists who came out with mathematical models to understand climate change. So this is correct. And yes, that's why it will also help in predicting climate change. It will help in the prediction of climate change. So that's also correct. So both the statements given here, both 1 and 2 are correct statements. Moving on, question number 18. Consider the following statements with respect to the 2021 Nobel Prize in Chemistry. They were awarded for the development of asymmetric organocatalysis. And then asymmetric organocatalysis is a novel technique of catalysis that is efficient, precise, cheap, fast and environmentally friendly. Alright, so please mark your answers. Let's have a discussion on it. All right, let's discuss this. So we are talking about asymmetric organocatalysis. Now organocatalysis is when you are trying to use an organic compound, an organic material for or as a catalyst. Now what is the role of a catalyst? Catalyst will help in increasing the speed of a reaction, right? So now most of these catalysts, if you observe, you'll find that most of the catalysts are metals. Most of the catalysts are metals. So here what we are doing is we are using organic compounds as catalysts. So that's why, yes, this was given, it was awarded for the development of asymmetric organocatalysis. And secondly, asymmetric organocatalysis is a novel technique of catalysis that is efficient, precise, cheap, fast and environmentally friendly. This is also correct, because this is one major problem that we have with metals, especially when most of these catalysts that we are using in any given reaction, they are mainly metals. And because of that, what we see is that these metals, they very often, they are very they have a lot of negative impact on the environment, on the entire setup. So they are poisonous in many cases, they are toxins. So that becomes a little challenging altogether. Also they are costly. So that becomes all these problems. There are all these problems with using the normal metals that we usually do when it comes to using metals as catalysts. But here what we are doing, we are trying to use organocatalysts, meaning that these are catalysts which are organic molecules which are easy to use. They are easy to use, they are cost effective, they are environmentally friendly at the same time. So that's why we see that in all these cases this is correct. So that's why both the statements given here are correct. Moving on, the last question of the day. Consider the following statements with respect to the 2021 Nobel Prize in Medicine. 1. Research has allowed us to understand how heat, cold and mechanical force can initiate nerve impulses. Second, this discovery could pave the way for new painkillers. So these are the two statements given. Mark your answers, please. Alright, so I think most of you have marked the answer and most of you have marked the answer correctly. So the answer here is both 1 and 2. So when it comes to this Nobel Prize in Medicine that was given, basically understand that this Nobel Prize was given for understanding or discovery of the receptors that we have for pain, the physical manifestation of pain, not the other one. So when we talk about this pain, whether due to temperature, whether due to heat, whether due to any mechanical force, whether due to anything. So any physical form of pain is something that we feel. All right. So you know that there are multiple forces can lead to pain, temperature can lead to pain, heat can lead to pain. There are so many things, right? So these are the receptors that they were able to discover. So there is the temperature and heat pain receptor, and then there is the touch pain receptor that we have. So overall, that's why we see that there are all these receptors these are the two basic receptors that were found, that were discovered. And because of this discovery, we see that we were able to understand how exactly does our body respond. What are the genes, what are the various cells that respond when it comes to the physical manifestation of pain. So that's why the other kind of pain cannot be called as pain. Alright, it cannot be called as pain. This is not the pain that we are talking about here. Now, the discovery of this pain receptors, what it does is that it also helps us understand how pain works. How do we feel pain? What exactly are the factors that help us in feeling of that pain? And how can we make painkillers against it? How can we make more effective painkillers against these kind of different forms of pain that we feel? So that's why we feel, that's why we see that in all these cases, this will help us in making better painkillers. So that is why when it comes to this particular question, both these statements given here, both the statements are correct, that it will help us making better painkillers, and it has allowed us to understand how heat, cold or mechanical forces can initiate nerve impulses. So this is about the last question, that is about the pain receptors that were discovered, and hence it will be very, very helpful when it comes to medicine of the future. And hence, of course, they were also awarded with the Nobel Prize. Alright, so this is it. This is it for today's session. So today we discussed three important areas. We discussed about biotechnology, we discussed about IT and computers, and then IT and communication, you can say, and then we discussed about the Nobel Prizes. We'll meet back tomorrow. Tomorrow there are three more important areas that we are going to discuss. We are going to discuss diseases, we are going to discuss defense, and then we are going to discuss energy. These are the three important areas that we are going to discuss in tomorrow's class. So all the questions, all the current affairs areas of last 1 year, or let's say 1.5 years, we'll discuss all of that in tomorrow's session. So until then, goodbye, thank you, good night. Please go back, revise well before you come back to the class again, and let's meet back tomorrow with a new set of questions again. Thank you very much.